Hi everyone. I welcome you all to the fifth lecture of course Cellular Microenvironment from Oxygen Gradient to Gene Expression Analysis. So in previous lecture, we gradually build our understanding of how cells respond to change in cellular microenvironment, and we first learned that uh, oxygen does not reach every cell equally because if uh, once the oxygen leaves the blood vessels or the capillary due to the diffusion as well as the consumptions by the cells, you will feel that if you are going far from the capillary there will be less oxygen, and that creates basically the oxygen gradient. And this gradient is being created within the tissue due to the consumption as well as the diffusion of the oxygen. We then explored how cells detect these decrease in oxygen through the hypoxia inducible factor or HIP signaling pathways, and that is one of the most important concept of this lecture series. We also learned that under normal condition, how HIF1 alpha is continuously get degraded by PSD enzyme and VHL enzyme. However, when oxygen level decreases, HIF1 alpha becomes stabilized, and we have already seen that. And uh, due to this accumulation of the HIF1 alpha inside the cells, finally it gets translocated inside the nucleus where it binds with the HIF1 beta, and that forms a transcription complex. And that transcription complex basically binds with the specific sequence of the DNA that is called as the hypoxia responsive element. Finally, we discussed why cell possess two oxygen sensitive transcription factor like HIF1 alpha and HIF2 alpha. Although these proteins have very similar structure and they regulate different group of genes, and that play distinct role during acute and chronic hypoxia. We have seen here. I'll discuss this in later on. Is that clear? So this creates an oxygen gradient within the tissue, and cells located further from the blood vessels experiences hypoxia. And we then explored how cells detect this decrease in oxygen through the hypoxia inducible factor or HIP signaling pathways, all of these things. And finally, we also discussed why cells possess two oxygen-sensitive transcription factor like HIF1 alpha and HIF2 alpha. Although these proteins have same similar structure or very similar structure, they regulate different groups of genes, and that they play distinct role during the acute and chronic hypoxia. Now, if you have been following this course carefully, you may have noticed that I have repeatedly asked a sentence or I have repeatedly asked a question that HIP activates gene expression or HIP turns certain genes on. But what exactly does that statement means? When we say that a gene is expressed, what is actually happening inside the cells? Does the DNA itself get changed, or does the cells produces a new gene, or in the informations already present inside the DNA? So these are some fundamental questions in molecular biology, and understanding them is essential before we can interpret techniques such as molecular biological techniques like RT-PCR or quantitative PCR, which we will discuss later in this course. So in order to answer these things, first we ask this question that what does gene expression means? And this is a highly important thing that is very fundamental concept, and it's a bit complicated. So I'll try my best to let you understand this concept without any fail. So when we talk about the gene expression, first we should know the definition because this is just a fundamental class. So I have written the definition here. So you can see that gene expression is the process through which the informations stored in the DNA is used to produce a functional product. That product may be your protein, or in some case, that is the functional RNA molecules. So this definition is very important. As we all know that DNA is the very stable molecules that carries the genetic informations and the whole genetic informations of the cells that has been stored in the DNA, right? And if that is, you can say that DNA serves as a main storage unit for the cells. And in subsequent slide, we'll see that in order to understand the gene expression, first we need to understand what is transcription, then translation. And in understanding these things, we will understand about DNA, mRNA, RNA polymerase, what is transcription, then what is translation, how this translation happens in the cytoplasm with the help of ribosome, and how this protein is getting formed. So we'll study many things in this lecture, and then we'll also understand how many types of RNAs are there, something like that. Okay. So now coming to the very fundamental unit, that is what is DNA, and in next lecture I will also discuss some more fundamental thing from chromosome to chromatin to DNA, something like that. Okay. But here, for this particular lecture, understanding of DNA is very important. So if we see DNA, DNA is made up of, you know, sugar phosphate sugar chain, and that is a double helix kind of a structure you can see, and both of these double helix get connected with the hydrogen bond through the base pair of the DNA. So we all know that there is a four base pair: adenine, thymine, guanine, and cytosine, and all of these bases are having some hydrogen bonding site, and due to this hydrogen bonding site, what happens, this gets binded with the hydrogen and this connect both of these strands of the DNA, and due to this hydrogen bond, DNA's molecules are very stable. And if you see the DNA, then the length of the DNA is very long. I'm not going to tell you these things right now. But if you stretch the DNA from our body, if you take all the DNA from our body, I mean, all the DNA from all the cells from our body, and if you stretch this, then you can reach from Earth to Sun 600 times. So this is how long this DNA is, but this is getting accumulated inside, or this is there inside our body. So definitely there would be something that would be there, that's why it becomes very small, and that it can get accommodated inside our body. So we'll definitely understand these things, how DNA structures looks inside our body, so that that much length of the DNA can be stored in our body, right? So we are all 99.9% alike, but if you see the humans, that is approximately 3 billion base pair of the DNAs are there in human genome. So it is itself a very large data set. And if we see the nucleic acid in our body, so there are two nucleic acid. There is the DNA, that is called as the deoxyribonucleic acid, RNA, that is called as the ribonucleic acid. So in DNA, the strands that we see here, that is made up of deoxyribose sugar, something like that. If you if you go in the details of the structure here, this is called as the deoxyribose sugar. And in RNA, that is the single, that is basically a single nucleotide sequence, that and that is made up of your ribose sugar, and these are the nucleobase base, that adenine, thymine, guanine, uracil, all of these things. But in RNA you will see uracil, but in place of thymine in the DNA, in RNA, you will see there is a uracil, right? So all of these are just a structure thing that you can read these fundamentals from any textbook. I am not going in the details of this right now. So the important thing in for this lecture is to understand what is gene. So as we have seen that DNA is the entire genetic, instructions manual, while a gene is a very one particular instruction within that manual. So if you see here, if I assume the whole this schematic is for the DNA, then you can see there is a specific sequence here that is known as the gene. And if I focus this gene part here, so this is again made up of various units like promoter, then there is a coding region, then there is a non-coding region, then there is a terminator. So the DNA strands is known as there are two kinds of DNA strands. There is a coding strands, there is non-coding strands, that I'll tell you, or there is coding and then there is a template strand, something like that. So we don't have to understand these things right now, but try to understand about the gene part. So as we have seen that there are various units that is associated with the gene. There is a promoter, and that is responsible for, that is also known as the regulatory region of the DNA. And this is located near the beginning of the gene. So if we see, this is the gene. So you can see this promoter is located at the beginning of a gene, and that acts like a docking site for the molecular machinery that starts transcription. So we'll see also that from here only RNA polymerase will come, and it will, it will start the transcription, right? Other regulatory unit here you can see in the DNA, that is the terminator. Once the polymerase basically convert RNA to, I mean, this template extends to the mRNA, then it will reach to the terminator region, and then RNA polymerase leave this extends, that is a template extend, and finally mRNA will leave from the nuclear ribosome. So that I'll definitely discuss later on. But here you can see there is a coding region that is called as the exons. Then there is a non-coding region which is called as the intron. So that contains the instruction sequence that are transcribed and after processing translated into the protein. So basically, uh, this is the region where you can see that the gene expression will happen, and this expressed gene will be transcribed into the RNA, and finally in ribosome it will get converted into the protein. So as we have seen that gene contains the various subunits, and all of these subunits are having different function. So when we talk about a gene is a specific region of the DNA. Obviously, we have seen this is a specific region of the DNA that contains the information that is required to produce a biological functional product. So that functional product is your mRNA or the protein. Then we have seen that functional product is mRNA and protein. Now we come to the HIF1A gene, let's say, for example. So that contains the instruction for the producing the HIF1 alpha protein. As we have seen that this coding region will contain some information, and that will get expressed over there. And then RNA polymerase basically converts this into mRNA. So for HIF1 gene that will produce a HIF1 alpha protein, ultimately EPAS1 gene that contains the instructions to produce HIF2 alpha protein, SLC244 genes that contains the instruction for producing the GLUT4 glucose transporter, and then VEGF-A gene that contains the instructions to produce vascular endothelial growth factor. So all of these genes are there, right? That is there in that all cells of the DNA. But again, that depends in which environment the cells are there, and what are the instructions or what are the genes that is getting expressed in that particular microenvironment, and what kind of expressed gene that should get transcribed into the mRNA for the formation of corresponding protein. Okay, so I hope the concept of gene is very clear to all of you, that we have seen that gene is a specific region in the DNA that contains the informations that is required to produce a functional biological product, and that biological product is your mRNA or protein. Now, next is that what does gene expression mean? So, gene expression means using the information that is contained in the gene to produce the functional product. So, as we have seen, that functional product was mRNA and protein. So, let's say for a protein coding gene express generally occur in two major stages. Like we will see, there is a transcription. So the transcription is the process in which the information that in the DNA is copied into the messenger RNA. So as I have told you, DNA is our master storage unit inside the cell that contains all the genetic information, but it should be stable. It should not get changed, right? Obviously, cells will try to preserve this stable information or actual genetic information. So cells are having some machinery that will copy this expressed information or information that is stored in the DNA, and then the copied unit will go for functioning of another thing. So that is the you can say that functionality of the cells does not want, or cells do not want to destroy their actual genetic information. It will try to preserve that genetic information, and in order to preserve that, if something has to do if cells has to do something, then definitely it will copy that information into the another form, and that another form is called as the mRNA. And again, the copy is being done by or the transcription has been done by the RNA polymerase. So here we can see that this is what the transcription is, where during transcription, what happens, the information in the DNA is copied into the messenger RNA using the RNA polymerase. RNA polymerase is an enzyme, and basically it does this kind of thing. So inside the DNA will remain inside the nucleus, but once mRNA will get formed after transcription, then this mRNA will get translocated outside the nucleus into the cytoplasm. And then what happens once it comes to the cytoplasm, let's say right now it is inside the nucleus. Now RNA will come to the cytoplasm outside the nucleus. And here basically what happens, this ribosomal RNA will come, and this ribosomal RNA, the tRNA, that is transfer RNA, that will translocate this mRNA to the cytoplasm. That is the function of the transfer RNA. And then the ribosomal RNA come, and then it will reach to the ribosome, where ribosome will start, reading this mRNA sequence. And ribosome basically read three base pair, and that three base pair will be called as the codon. And for three base pair, for one codon, there will be a specific amino acid, right? So now this ribosome, as I have told you, that will read three specific base pair of the DNA, that is called as the codon. And one codon will for one codon there will be one amino acid, and then another codon there will be another amino acid, and finally this in ribosome you will see there is a series of amino acid or sequence of amino acid. And we know that the combination of or sequence of amino acid is called as the protein. And finally this process is called as the translation, where this mRNA ribosome use the informations contained in the mRNA, and that will get translated into the functional protein. So this is how the transcription and translation process occur, and this is known as the central diagamma in molecular biology, where DNA can get mRNA gets converted from the DNA, and then mRNA will serve as a template for producing the protein in the ribosome. So this is all about how what does gene expression mean. So if gene expression does not happen, then what will happen? That expressed gene cannot get transcribed into the mRNA, and then cell will not understand what protein that is required. So in order to form that protein, that is the functional product actually. So then gene expression has to occur. Is that clear? I hope this is clear to all of you. I tried my best to let you understand, but again, this is the superficial thing. In subsequent lecture, again, we'll go in the deeper and deeper concept. We'll actually understand what is happening at the time of RNA polymerase, how RNA polymerase is, transcribing this informations in the DNA to the mRNA. Okay. So as I have told you, the overall process is called as the central diagram of molecular biology, where DNA is getting converted into RNA, and then RNA is getting converted into the protein, that is called as a functional product. Now coming to the next part, why are some genes on and while other are off? So as I have told you that this is one of the most important question in the cell biology. So when we see almost every cell in the human body that contains essentially the same DNA, like liver cells, muscle cells, nerve cells, all of these cells contains the same gene. Yet these cells look different and that performs different, functions. But the question is why? Because if we see different cell express different group of genes, like muscle cells expresses gene that is required for the muscle contraction. A nerve cells expresses gene that is required for the electrical signaling. A liver cell expresses gene that is required that is involved in the metabolism and detoxification. And a skin cell that expresses gene that is required for the barrier protection and repair. So as we have seen that the functionality of all the cells are different. So let's say muscle cells will express the gene only when there is a muscle contraction and expression, and then the expressed gene now will get transcribed into the mRNA, and now the ribosome will form the protein from that is inscribed transcribed mRNA that will be responsible for correcting the muscles, right? Like muscle, whether the correction is required, contraction is required, or expansion is required. Next, coming to in case of liver cell, let's say in metabolism or detoxification. So the cells DNA will express that kind of genes that is required for the metabolism or detoxification. Now that gene will get transcribed into your mRNA, and now the ribosome will read that mRNA sequence. Okay, now these cells require some modification or some kind of protein that will stabilize the metabolism or detoxification. Now cells will ribosome will form that kind of protein only, right? So first there should be some signal for the formation of protein, and that signal will is there inside the DNA as in the form of gene, and that already that signal got expressed. That is called as the gene expression. Means if gene expression has happened in the DNA, that means there is some signal that is coming from the DNA that will that signal will get transcribed into mRNA. Now mRNA will get translocated outside the nucleus with the help of tRNA, and then in ribosome this mRNA will ribosome will read the informations in the mRNA, and that will convert into the protein. So this is all about this thing. So now the concept is very clear that why are some genes on while other are off in different cells. So if we see here that the same DNA, but different genes are active in the different cells. So let's say we have DNA, all of these cells, all cells have same kind of DNA. But let's say in the muscle cells, gene A, gene B, and gene D are expressed, but gene C has not been expressed. In nerve cell, let's say gene A, gene C, and gene D is expressed, gene B is not expressed. Again, in liver cell, you will see gene B and gene C. This is just for example. And for D, you can see for a skin cell, there is gene B and gene D is getting expressed, something like that. So if you see that the expressed gene will give different informations to, or all these expressed gene will get transcribed into the mRNA. And now this mRNA will give different informations for the cells to the ribosome. So with respect to the muscle cells expressed gene, now the ribosome will form this kind of protein that will be responsible for muscle contraction or expansion. Then the nerve cells gene, the gene that is getting expressed in the nerve cells, now it will get transcribed into the RNA. Now ribosome will read this RNA, and then that will produce that kind of protein that will be responsible for the electrical signaling, and so forth, something like that. So this is the importance of the gene expression, and this is very important. So here we have seen that gene C for muscle cells, it does not require expression of the gene C, because that does not lead to the any muscle contraction or expansion. That's why this gene is not getting expressed, right? That's why this gene is turned off. But if gene A and gene B and gene D is required, it is required to express because of muscle contraction and expansion. That's why this gene got expressed in this, and now this will get transcribed into the mRNA, and finally it will go to the ribosome for the protein formation or corresponding protein formation. Is that clear? So that is why some genes are getting expressed in a cell, some genes are not getting expressed in the cell. But if you'll see, all these genes are there in all the cells, but all these genes are not getting expressed. Different genes are getting expressed in the different cell because the working function of all the cells are different. Now coming to the next thing, what does it mean when a gene is on and off? So as I have told you, that is the same function. This, if gene is turned on, then it will get transcribed into the mRNA, and finally it will get translocated into the ribosome for the formation of the corresponding protein. So a gene may be completely inactive or weakly expressed or moderately expressed or strongly expressed. That is the importance of a gene expression. So here I'll stop this lecture here, but uh, in order to summarize this lecture, what we have learned in this lecture, basically we learned the central diagram of molecular biology, where we have seen that the express in the DNA is getting transcribed into mRNA, and then this mRNA gets translocated outside the nucleus with the help of transfer RNA, and then this mRNA will be, read by ribosome to convert this into the functional protein. So I'll stop this lecture here. In the next lecture, I'll do discussion about how does a cell turns gene on or off. So this is another important part, and this is again we are going in the deeper and deeper of this concept. As I have told you, the motivation of this course is to let you all the concept, to let you understand all these concept in the well detailed way. Okay, so I'll stop this lecture here. See you in the next lecture.